Hello. So let's do this cloning. I will share my screen. All right, here we go. So now you should see the SnapGene. So I assume that you managed to install the program and that you are able to open it. I have to warn you that there are some bugs in this program, and some of these bugs actually are even preventing you from opening some files. So if you have some technical issues, get into contact with me. I actually have submitted even a bug report last week concerning some issues like with opening some specific files. Okay, but let's get going. So of course, I have been showing you during the lecture that normally what you do is the most simple type of cloning is you have one vector and you have another vector, plasmids typically, and you want to subclone some piece of DNA from one place, here this donor vector, then here into this other vector, into this acceptor vector. So meaning that you are starting from two plasmids. And so the first thing is, of course, where do we get these plasmids from? And getting the first plasmid actually is relatively easy. So because you can download it, of course, from the Moodle page. So let's do that. So here is the Moodle page. So this is the so-called donor vector. So this is where we are taking our DNA piece from. So let's save it here. So now we have here downloaded on our hard drive here this donor vector where we will take this VGFC sequence from. Where do we get the other vector from? So, and I wrote in the descriptions that you want to use another plasmid, and the name of the other plasmid is pcDNA 3.1 plus. And there are many ways how you can get to it. I mean, a typical way would be to Google it and to find the sequence somewhere. But SnapGene actually makes it very easy for you, because you have here under the file option import, and then you have many different options. And I can't tell you, it's very difficult to know which one of the options is the correct one to choose from if you're looking for a specific vector. But SnapGene Online Sequences is always a good way to start, because that's actually a database which the producers of the software are maintaining. And of course, it's in their interest to have as many plasmids there as possible to make it easy for the user. And so we are just going to search here for this plasmid pcDNA 3.1, and you can see that there are many different pcDNA 3.1 vectors, and we want to have the top one here, 3.1 plus, and we just push import, and now we have the target vector where we want to clone our sequence into. So then we still have to open our donor vector, and now we have both inserts, both plasmids here in our SnapGene, the PREP7, and here this is the gene that we need to subclone into the other vector, and this is the acceptor vector. And if you remember correctly, so the idea is, of course, to use restriction enzyme cloning, and we identify here the multiple cloning site where we have lots of restriction enzymes. And then, of course, here we have our gene that we want to subclone, and we have to identify restriction enzymes on both sides that we can use to do this subcloning. And you can see here the restriction enzyme sites. If a restriction enzyme site is in bold, then it means it's only once present in the plasmid. And these are actually the ones that you typically are interested in, the so-called single cutter enzymes. And the only thing what you have to do is to check, like, do you find here one of the enzymes, which you can find here also in the multiple cloning site. And of course, the important thing is that you want to clone the gene into the correct direction. So here you have the promoter, and the direction goes this way, and the gene, of course, goes into the same way. And we have to maintain this orientation. Otherwise, the gene is not going to be expressed. And we can have a look here. We have the promoter here in the same direction. So we have to actually make sure that this gene goes into the same direction here. And so this is why you have to look at sites which are upstream of the coding sequence and downstream of the coding sequence. And they have to be also here upstream, meaning here on this side or on this side. And one possibility, I mean, there are many possibilities, and many of you have actually chosen different possibilities how to do the subcloning. And I will just show one possibility, like probably the easiest if I choose hint 3 here and not 1. Both of them are very commonly used enzymes. So I select hint and then I push down the shift button and I select not. So now I have selected here the fragment, which is excised by using these 2 enzymes. And then I go to the acceptor or to the target vector, and I choose exactly the same enzymes, hint and not. And again, if you push hint and then push down the shift key and then select not, then you will have both of them selected. And then you just go to the actions menu, restriction and insertion cloning, and insert fragment. And the vector is already here prepared, it's cut. And up here you have the different tabs, and we just move over to the fragment tab and select the plasmid from which we want to cut out our insert. So this is our donor vector, and we select the same 2 restriction enzymes, hint and not. And then you can see that the fragment shows up here down. It's ligated here. This one is shown in green. No error messages are displayed. And the clone button is enabled. So you can just push the button and here you have the ready construct. You should, once you have done this cloning operation, save it under some name, like here pcDNA3. Human VEGFC full-length wild type would be a nice name, because once you have done a couple of clonings, you will just get confused if you don't have a nice naming scheme that shows you exactly what you are dealing with. And now you can look here. You have here the map. You can look at the sequence in detail, but very importantly, you have the history. So basically everything that you did, meaning like starting from 2 different plasmids and then cutting out here this red piece and with these 2 enzymes, and then replacing here this tiny piece, hint not, in this plasmid, and then inserting it as a fragment and then getting the new one. Everything is maintained in the history. And this is important because you want to have always, so you don't really need the original files anymore. You can just close them. And once you have saved this one in this new file, you will have the full sequences of these originating plasmids together with the way how you did the subcloning. And that is important. You want to have one file, one single file, which basically describes everything what you did in the lab. So if you have 2 files, that means you have been doing something wrong. The same, of course, if you have one file, but you don't have a history, then you have also done something wrong, because other people are not able to replicate what you have been doing if you don't have the history. You can actually manually delete the history, but you should never really do so. So hint and not is not the only way how you can get the cloning done. So let's get the cloning done using something else. So I have to open again here the import again from the SnapGene database, the pcDNA 3.1. And so what else? What other kind of, now let's close this one. We don't need that. So what other kind of cloning options do you have? You can simply use different enzymes. Let's use BMT and XHO here to cut out our fragment. And then we could go here and then do the cloning restriction insert fragment. Okay. Now I have selected the wrong one. We have to, oh no, it's actually the correct one. So up here, so what did we use to BMT we wanted to use here? And here, for example, we could use PSPX1 as a restriction enzyme. So this would be, we would cut our vector with these two enzymes, and then we would choose our insert from here, BMT and XH01. And you can see that again, this shows up in green, meaning that the cloning is feasible. And down here, you can see what's really happening. And as you can see, actually, the interesting thing is we have been cleaving our vector with a different enzyme than the fragment here. So we have been generating the vector with a different enzyme than we used for the fragment. Here we used XHO and here we used PSPX1. It doesn't really matter. The only important thing that matters that those two enzymes, they produce the same overhangs, because that is the only thing that counts if you want to ligate those two ones. So you can use different enzymes if they produce the same overhang. You can obviously here, instead of the BMT1, XHO1, also use the same restriction enzyme here. It's still possible to do the cloning. All right, so this would be another way how to do the cloning. However, some people of you, they have probably being used from doing so from Microsoft Word or any other text editor, have been doing a cloning, which is definitely possible because the program SnapGene allows you to do that using Control C and Control V, meaning to copy and paste, like you do copy and paste in the text processor. And that works. The program allows you to do this. The problem is that you usually do mistakes when you do that, because it's actually quite difficult to do that. Let's see if I select here. Okay, let's get rid of this one and let's start here. So this is the stuff that actually we are really interested in. And we used actually these enzymes from hint to here, and we cloned actually some sequences that we are not really actually interested in. This is the only thing that counts for us. So can you just select it, then hit Control C, and then go here, and then have a look here somewhere at the sequence, somewhere in the multiple cloning side, go somewhere there, and then hit the Control V button or just say paste. Yeah, you can simply do that. So the program allows you to do this. Now we have done that. The problem is you cannot do it in the lab. And the reason, of course, why you cannot do it in the lab is that, yeah, there is no way really to cut the DNA exactly at this position where you have been doing the cutting and the pasting. So it doesn't really work. So never use cut and paste in order to do a cloning step in this program, but only use the predefined actions. Because here under the predefined actions, there are the things which you can really execute in real life in the lab. It is actually possible to do with using Control C and Control V with cutting and pasting correct clonings, but you have to be extremely careful. So let's just undo this. So how would you do this? Actually, it's very difficult to do it from here to here because there are simply no enzymes. We can try to find additional enzymes. Like here, now we have more enzymes. Here we have actually one enzyme which might do the trick. And here, but let's say I select now from here to here, and I do not want to use this restriction and insert cloning. I mean, you should, but just in order to show you can do it correctly also with Control C and Control P. So if I now do here this cutting with a Control C, the important thing is I really have to know where I'm cutting and where I'm inserting. And here, this, of course, would already not work because I have been selecting the hint enzyme here and the STU1 enzyme here. So let's see whether we can do, actually it would work. Now I have pushed this copy restriction fragment, Control C. And now we go here to that and we choose, what was it? Hint, and I have to choose now something like that fits. For example, echo RV would fit with this, and then I hit Control V, and now I have it done. The interesting thing is, if you do it and you do it correctly, the program actually realizes that you do it correctly, and it pulls up exactly the same window that is used when you actually go to actions and insert restriction and insert fragment cloning. So, and then of course the only thing that you have to do is to push this clone, and now you have done it correctly. But never do Control C, Control V. So if you don't see the real dialog window appearing, which is responsible for this restriction fragment cloning, so then you are probably doing something wrong. All right. Still something about selecting restriction enzymes. Some people have been using like this hint. Some people have been using the BMT. Some people, there are so many different possibilities. How do you choose a good one? Not all restriction enzymes are created equal. And so the rule of thumb is, I mean, of course, use the restriction enzymes that you have in your fridge. Because even though you might be able to do the restriction enzyme cloning in the within the SnapGene software, if you don't have the restriction enzyme in your fridge, you can't do it in the lab. So this is number one rule: only use restriction enzymes that you have. Number two rule: if you don't have restriction enzymes and you have to buy some, then try to use restriction enzymes that are very commonly used, because these will be cheap. So there are big differences in the price between different restriction enzymes, and the thumb rule is the more frequently and the more people are using them, the cheaper they are. So restriction enzymes like echo R1 and hint3 are very cheap. BMT is a less used one, so it is more expensive. And the difference, for example, between, I think, echo R1 and BMT, it's about 5-fold, and there are some other even more rarely used restriction enzymes where the difference can be even bigger. And of course, if you use a frequently used restriction enzyme, they are not only cheap, but you also will typically find them from the fridge. So this is just a sample. How do you know? Of course, if you haven't done any cloning, you don't know what are frequently used enzymes and what are not frequently used enzymes. One way how to figure it out is go download a price list from New England Biolabs and then look at the prices that you are paying for the enzyme. The less you are paying for the enzyme, the more frequently it's typically used. But be careful. They are sold in different concentrations and in different volumes. And in reality, you also have to look, of course, at the unit definition of the enzyme. So it's not very trivial, even based on the price list, to figure out whether you have a frequently used enzyme or not. All right. Still one thing, of course, if you end up with the history, you always want to have, of course, 2 starting plasmid vectors, because it's very seldom that you have it happens, of course, if you make synthetic DNA, you can, of course, start from a linear vector, but this is typically the exception, especially when we are dealing with the so-called subcloning, which means we are starting from 2 plasmids. So you don't want to have here anything that is a linear fragment, because you don't really have linear fragments in your freezer. So the other thing, of course, what you don't want to have, you don't want to erase your history, because then I'm not able to evaluate your cloning operation. So if you send me, if you do this erase cloning operation, you still have actually the resulting plasmid, but I cannot tell you whether what you did is actually feasible in the lab or not. So if you don't have any cloning history in your SnapGene file, then please redo it again to record the cloning history and send it to me. All right, I guess that is all what I wanted to tell you. But if you have any problems with the program, despite this short demonstration, please get into contact, because we can sort them all out. And some of them are unfortunately non-trivial, especially those when you have, for one or the other reason, run into one of these bugs. And these bugs actually occur in, this is independent from you. It has actually something to do with the remote disks, if they are mounted on your computer or not. If they are, with some certain remote disks are mounted on your computer and you open the program, then you won't be able to open certain files. So don't hesitate to contact me, and we can sort it out also in person. Okay, thanks.